Vakode sir is presently working as professor at Delhi Institute of Pharmaceutical Sciences Research, DIPSAR, a custodian college of DPSRU, Government of City of Delhi. He has completed his PhD in the pharmacy from the Department of Pharmacy, SGS Indore, under RGP Bhopal, and M.Pharm from Birla Institute of Technology, Mesra, Ranchi. Sir has received Young Scientist Award grant under Fast Track scheme from the Department of Science and Technology DST. His research interest includes designing, planning, and synthesizing new organic molecules of therapeutic interest and its biological evaluation. He has supervised many of the PhD students and many of the AMFOM students also. He has more than 75 publications in international and national journals and has more than 25 years of teaching experience. Prior to joining DIPSA through UPSC in 2001, he worked with the Bokar Limited and Vidyabharati College of Pharmacy, Amravati. He is also associated with various professional associations like APTI, IPGA, SPER, APP, APSE, and he delivered a number of guest lectures, chaired many sessions in various international and national conferences. Sir is expert in various committees of AICTE, PCI, as well as Ayush. He is also the member of Board of Studies for Central University of Rajasthan, SGP Amravati University, and IGNTU Amarkantak. So I request Sir to kindly start the session, sir. We are very fortunate to have you here.
Uh,
Over to you, sir.
Priyanka ma'am, sorry, Sarah sir, I just have to make an announcement. Many of the participants, they have joined meeting in some other name like FDP 11144 and like that. So their attendance could not be marked. Please rename yourself, otherwise we would have to throw you out from the meeting. Please. Sir, I am very sorry. Due to this attendance, I have to announce this. Now you can continue, sir. Sir, welcome, sir.
Thank you, Madam Priyanka, for detailed introduction. I'm not that much a great person or a very senior person. I'm like you people only, and it's Dr. Arun's request, which I cannot deny it. He's a good friend of mine, and it's a honor for me also to interact with all of you and to meet you people over here in this platform. And first of all, I should congratulate Dr. Arun Gupta and his team for taking lead in arranging such a nice program with such a good title, Artificial Intelligence and its utilization in the pharma field. Okay, and which is definitely a need of an hour. Okay, so even all of us, most of us are not good or experts in the field of this computationals or artificial intelligence, but in today's scenario, what we are observing is that we have to come to the track. Okay, जो growth का track हम लोगों ने I'll be using Hindi as well as English also. I hope all of you must be aware of this thing. Okay, no problem. So we are in this very developing stage. Earlier we were saying that India is developing nation, but now we are meeting at par with the developed nations. Okay, and especially in the field of pharmaceuticals and medicals, okay, medicines, we are leading the world, and this present pandemic has shown a way that yes, India is going to be a superpower. Okay, not in all respect, but at least in the pharma field, we are going to be definitely a superpower, and that India has shown the way to the world how we can deal with the such type of situations where all very developed nations and who has got a very sophisticated, you can say that their infrastructure was very sophisticated. Okay, and then too they were unable to deal with this type of condition, but India could make out, and we have already handled this solution situations very nicely, and I hope with the help of all of you, the distinguished faculty members and the scientists from the pharma field, will be definitely able to conquer this COVID-19. Okay, and I expect, I request again, I humbly request all of you to spread the awareness again with the family members, friends, students, because as a teacher, we have got a moral responsibility to, because there are soldiers who are fighting on the फील्ड और दी बॉर्डर्स, चाइना बॉर्डर पे तो वो लड़ रहे हैं, बट हमें अंदर का जो एनिमीज हमारे इसमें है अभी प्रेजेंटली, सबसे मोस्ट डिफिकल्ट और जो खतरनाक एनिमी हमारे लिए है, दैट इज कोविड-19. सो वी हैव टू डिफीट दैट एनिमी, एंड एज अ सोल्जर ओनली हमें उस, एज अ एजुकेटर, फार्मा एजुकेटर, वी हैव टू dealt with this problem. Okay, so as we have seen that this program is basically on the artificial intelligence. Okay, so in the pharma or medical field, actually I'm not at all expert in the artificial intelligence. I'm basically B.Pharm, M.Pharm, PhD in pharmaceutical chemistry, and I'm fortunate that I'm PhD from SGSITS Indore, RGPV Bhopal, and Dr. Arun Gupta was my colleague when we were doing the PhD. Okay, so whenever we go for this type of higher educations, especially research-based things, everywhere we need to go for the biological activity, pharmacological activity, and at one point, something somewhere we get stuck. Same thing happened with me also. That while doing my PhD work, the type of facilities which was required, that was not very easily possible in SGSITS Indore, because pharmacology department that time professor was just resigned from there, वहां से निकल गए थे, और दूसरे प्रोफेसर फार्माकोलॉजी के लिए इतना ये difficulty था, and we were not having sufficient very good type of animal house that time. Okay, I hope they must have developed it now, but it is mandatory requirement now in PCI, AICTE, everywhere, that animal house must be there. Okay, and whenever we are writing certain projects, especially at DST, DBT, and ICMR, whenever you are writing a project to the ICMR, they will first give you the thing, yes, what is your protocol, institutional ethical committee से आपने पास करवाया कि नहीं आपका protocol, whether you got clearance from the institutional annual ethics committee or not, and there the things get stuck up, because institutional annual ethical committee meeting will be there in 6 month, once in a 6 month, or once in a 1 year somewhere. Okay, and the problems or the projects get stranded because of that. And from that point of view, there are n number of difficulties researchers were facing. Okay, so if we consider the drug discovery process, the new drug or new lead compound therapeutic target is a very complicated and very important steps. Lot of steps are there: drug design, synthesis, and lastly comes the pharmacological screening. Okay, so in that case, drug development stages, we can say that basically 3 stages are there. Stage 1 is the hit and lead compound development phase, preclinical studies, especially in vitro as well as in vivo studies, and stage 3 is the clinical studies. So last stage is very, very important. So everywhere we are seeing the news of clinical trials, क्योंकि वैक्सीन की क्लिनिकल ट्रायल्स बची हुई है, जो और कैंडिडेट्स हमारे बचे हुए हैं इंडिया की जो शायद डेवलप किया है वैक्सीन, तो एवरीथिंग it was delayed, vaccine was delayed because of the clinical stage, preclinical stage, clinical stage. Okay, so all the country or all the you can say total world has discovered or mainly come to know the power of the pharmaceutical scientist, pharmaceutical industry, especially in this pandemic. Okay, and all of a sudden our DCGI came and announced that yes, we have finished all the thing, and the vaccine Covishield is launched for use in the human being. Another vaccine they have given conditional acceptance, and it's going to be marketed very soon. Third, another vaccine by the Cadila is also in the pipeline, and so many vaccines are again in pipeline. Okay, so these are the stages, and whenever we want to go for preclinical studies or clinical studies, we need to study the animal animal uh, models or experimental models we need to study first. Okay, so in that case, where these animals are used, especially in case of pharmaceutical institutes, okay, we are doing it for the biomedical research, okay, for studying the body system, disease processes, health, to develop the vaccines and new treatment for the diseases. Okay, so in the initial stages, when we were at 10+2 or diploma level or at degree level, we used to dissect the earthworm, cockroach, okay, so frog. That was possible, but afterwards, because of the stringent norms by the CPCACA as well as the stringent actions taken by the animal welfare people like PETA and that type of organizations, it has gone, or we can say that it has vanished at the diploma level, undergraduate levels. Okay, and only demonstrations were possible. At PG level, they are allowing it with restrictions. So product safety and testing for that purpose, once the product is formed, okay, it has to be tested first on the animals. Okay, like any type of cosmetics, households, cleaning products, pesticides, especially pesticides require too much use of the animals. And lastly, what we are dealing is the education. Okay, for the especially pharma students, the veterinary students, medical students, uh, to study the behavioral and biology of the animals. These animal experiments are carried out, and n number of animals. You can see that in this is old figure of 2017, 4.51 million was animals were used, and so many different species like mice, rats, fish, birds, pigs, guinea pigs, especially. Okay, goat, sheep, cattle, guinea pig, rabbits, okay, dogs, the higher animals, and primates like monkeys. Okay, so these type of animals were used in the drug discovery. Okay, and animals will suffer needlessly in such type of science. What we are saying that. So there was a very hue and cry from the animal welfare department that animals well-being should be protected. Okay, whenever you are conducting any type of research activity, biological activity, it should be free from the hunger and thirst, especially for the animals. There should be freedom from discomfort. Animals should not have a discomfort. Freedom from pain and injury and diseases. Freedom to express normal behavior. Freedom from fear and distress. Okay, so all these things were we are supposed to understand the importance of all these points before carrying out the animal experimentation. Okay, so this was very difficult for the researchers. So there were alternatives to the animal experimentation. So on this topic, I used to deliver earlier lectures, but in present scenario, we have to go further also. Okay, so in case of animal alternative to animal experimentations, we have, it has been mentioned that there are 3 R's, especially replacement, the reduction, okay, and refinement. These 3 R's was mentioned in the books, and in some literature, we are observing that we need to go for the 4th R, that is rehabilitation. Okay, that is once the animal is used, after that rehabilitation. And fifth are what I am proposing, and so many scientists are also proposing, that is the reuse of the animals. So whenever possible, we should try to replace the animal model. Okay, and here we can use in vitro techniques with cells or tissue cultures, replacing it with the higher animals with lower animals. Okay, especially microorganisms whenever possible, plants or eggs or amphibians whenever possible. Then live animals may be replaced with the non-animal models such as dummies or computer modeling, and use of AI is here. Okay, so we should try to replace the animal models whenever possible. If not possible, the next important thing comes the reduction. Number of animals should be reduced, because if whenever you are going to any in front of any ethical committee, the most important concept will be how to minimize the number of animals. सबसे पहले उनका रोल एनिमल एक्टिविस्ट का हो या सीपीसीए मेंबर का वही रहता है कि आपका प्रोटोकॉल है, that they will see that how it will not be cleared very easily. आपका प्रोटोकॉल इतना इजीली क्लियर नहीं होना चाहिए, यही उनका सबसे पहले रोल रहता है. Okay, so in that case what happens, they will always ask questions, so many queries. आपने ये परफॉर्म किया है, are you expert in this, are you, will you be able to give this thing, will you be able to withdraw the blood from properly, and why you need this much of animals, reduce it, reduces, we cannot give it. Okay, so during our MPharm or PhD projects especially, we used to use 100 200 animals, but now especially in our institution, our ethical committee experts will not allow more than 50 60 animals in one go. First finish that, show the results, and then go for further experimentation. Okay, so reduction is the important thing which is required in the alternatives to the animal experimentation. Okay, so how we can reduce it? Sound experimental design. So experimental design should be proper one with the proper expertise used. Okay, then retrospective analysis. Kitna lagega, how much is required, how much is animals are specifically required for the statistical analysis. Then this pilot studies, we should first study that yes, where, how we are going to do the things. Then ethical review from our side, internal side, we should also ask, yes, whether what I have demanded is ethical. Okay, and use of reuse of the animals. If washout period is there, and after washout period we can reuse the animals. Then harmonization of the guidelines. So we should try to follow the all the guidelines. Okay, and third R is the refinement. Refinement means the experimental protocols. So we should, whenever the things are there that we are studying the some Ayurvedic formulations, so Ayurvedic expert physicians should be there in your experimentation. If you are going for some radio labeling or a radioactivity related protocol, then the scientist from that respective field should be part of your studies. Okay, so that he will refine it. गलती होने के चांसेस ना के बराबर होने चाहिए वहां पे भी. जहां हम देखते हैं कि हम जब किसी हॉस्पिटल में जाते हैं या whenever we go to any doctor, we expect that yes, आज जैसे मैंने given the blood samples, I ensure that yes, the person is DMLT minimum and he has got sterile all the needles, disposable things, he is using the disposable gloves. Okay, या जहां भी हॉस्पिटल में जा रहे हैं, we are saying that yes, it must be most perfect way which you should be treated by that person, doctor or जो भी है technician, he should use the proper protocol. So in same way, if we are handling the animals, our experimental animals also need that yes, your protocol is refined one. Okay, and next fourth R is the rehabilitation. So once the experiment is over and the animals are not required to be killed during experimentation, then they should be rehabilitated properly. Okay, and after washout period, fifth are reuse, especially even if uh, after washout period we can use it, especially for the UG demonstrations and other purposes. And in some cases, what we are doing, we are using heart of that particular frog. Okay, and once you remove the heart, the other muscles are there, so you can use that muscles to show the demonstration or any experiment on that abdominal muscles of that frog. So reuse simultaneously, यहाँ पे हार्ट तो आपने ले लिया, एक स्टूडेंट्स या एक ग्रुप आपका हार्ट पर कर रहा है, another group will go on to the that muscles, एसिटिलकोलिन का इफेक्ट क्या आ रहा है, बाकी और चीजों का क्या इफेक्ट आ रहा है. Okay, so that thing can be done in case of reuse, so that we'll get the maximum output from the animal experimentation. So what is the need of an alternatives? Okay, there are so many legislative authorities, especially in India. Okay, CPCACA is very harsh, okay, in implementing its rules regulations, and the animal activists from the PETA, especially Delhi or metro cities, you will say that जब भी आपके experimentation कुछ चल रहा हो, they will come, yes, how we are doing it, have you got the permissions, nahi hai, then even legal actions can be taken against the researcher or the specially directors जिनके अंडर में animal house है और who is holding all this capacity. So that is why it is always better to go for alternatives whenever possible. Okay, जहाँ specially PhD work है, कुछ M.Pharm work है, then we need to go for the animal activities. And there are lot many agencies throughout USA, Japan, EU, European Union. So they are form this type of ICATM guidelines. Okay, and through that ICH guidelines came, OECD guidelines came, ICCR guidelines came. Okay, regarding the testings of animals, alternatives to the animals. Okay, and as we are seeing that total world leaders, world uh, leading countries are involved in this biological activity, because everybody want to show the progress, highest progress in the field of medical research. Okay, so these guidelines are framed, and if we see, there are so many other alternatives also, epidemiological studies. So there are various products, like epidemiological studies reveal that smoking is associated with the lung cancer. Okay, so in that case, pardon, Arun, human skin equivalent system. Okay, so epiderm or skin ethic that we can see here, uh, epi skin type of things can be there, that scientists are developing a skin which is mimicking the structure as that of the skin, and where we can avoid the use of animals. Okay, there are so many things available. Human cell lines as well as tissue cultures are also available in the present scenario, especially for the breast cancer treatment. The human dopaminergic neurons are used. Okay, here in Parkinson's disease, and especially whenever like people like us, synthetic chemistry people, we synthesize the compounds. Okay, we analyze it with the IR NMR mass. Yes, this is form. We perform the structure activity relationship. Okay, drug design part, and finally once the compound is confirmed that yes, this type of desired compound has been formed, we send it for the biological activity in our department itself, or if not possible, then we send it to the institutes where the cell line facility is available. So we generally use to the uh, IIM Jammu, okay, where my student itself is scientist and working in this cancer cell lines especially. So we send these samples to them, and they can easily find out whether our chemical compound is effective against this type of cancer or not. Okay, So this is the simple thing. So which can give you clear idea than what we will get with the animal activity even. So this should be done at whenever possible. And after that comes the new generation thing, new concepts that in silico predictors are also available. So virtual tox lab, kick prop, pharma expert, okay. And there are so many drug design QSR softwares, especially Schrodinger, okay. They are also, we are getting such type of information to some extent from this, and so many good computer softwares are getting developed day by day, okay, which will mimic the human system, okay, and which will mimic the some pharmacological aspects of that particular animal species also. So we can avoid animals and use these softwares, okay. So non-animal computer simulators are also there. Battlefield induced traumas can be there. Trauma man is the Kaitosan films. These are the certain alternatives to the animal experimentations. So what are these benefits? So these are more reliable than the animal tests. Okay. So animals test, why it is more reliable? Because each and every person, आप देखते होंगे, even at our home itself, कि हमारे घर का खाना अगर कोई दूसरा बना लेता है तो उसकी टेस्ट बदल जाती है. आप डिफरेंट डिफरेंट रेस्टोरेंट्स में जाते हैं बिकॉज ऑफ इट्स टेस्ट. स्पेसिफिक इफ यू वांट टू गो फॉर, नहीं नहीं ये साव जी का ये डिश अच्छी है. इंदौर में आप जाते हैं तो कोई, हाँ नहीं नहीं ये दिस इस होटल का ये वाला पार्ट बड़ा अच्छा है. छप्पन भोग में भी आप मार्केट में जाते हो कहीं पे, ओके, यू विल सी दैट यस ये जोशी जी के वड़े that is famous for that. So it varies from individual person to person, and because of, so along with science there is art in performing all these activities. So same way in animal experimentation, although we are doing with the same type of animal, same type of test, slight variations are possible because what type of animal we are using, where you have stored that animals, आपका animal house ढंग सा ये है कि नहीं, whether the animals are stressed या stress free है कि नहीं, okay, whether we are handling them properly or not. आप handle तो ढंग से कर रहे हैं, but साइड एनिमल हाउस के साइड में कोई बिल्डिंग कंस्ट्रक्शन हो रहा है एंड दे आर यूजिंग दी स्टोन क्रशर या टाइल कट करने का आवाज जो आप देखते हो, सो गिनी पिग्स आर वेरी सेंसिटिव, दे विल चेंज देयर फार्माकोलॉजी, उनका काफी सारा बिहेवियरल चेंजेस आ जाएंगे उनमें. So you will not get a good result with such type of animals, okay. So reliability becomes difficult, okay, sometimes if we are not having a very good animal house with you, okay, and more cost effective than the practical. Guinea pigs, maintenance of these golden hamsters, okay, is very very difficult. Even maintenance of animal house itself is very difficult. Very few good organizations and government organizations are easily possible for them to maintain the animal house. Otherwise the requirements are so stringent that it becomes a very costly affair to carry out the animal experimentation, and that is why these alternatives should be used, and we should go for the softwares like biolab, pic, cd-rom. This is also available. There are so many other alternative softwares which can be uh, give us the total ability that we are performing a type of dissection. Okay, even आप बच्चों के देख रहे होंगे कि उनके मोबाइल पे जो गेम्स चलते हैं, okay, they can see that how somebody is hiding behind that, और कौन से गेम नए नए कि इवन गवर्नमेंट को अलर्ट होना पड़ रहा है कि यस दिस गेम्स शुड बी अवॉइडेड, इनको हटाना है, okay, because that is creating some psychological problems in the children especially, okay. So that type of things are there, and that is totally based on this type of concepts, uh, and especially machine learning and AI based type of games are also available. Okay, so आप छोटे देखते होंगे कि बच्चों को भी डॉक्टर बनने का भी शौक हो जाता है, so they will go and perform the surgery on the human being. And same way, animal experimentation, we can do it very easily. Dissection, we can do it very easily with the help of these softwares. So X-Farm Tutor was one of such important software which was developed. One of our friend, Dr. C.R. Patil, who is now professor with us, also discovered this type of software, who is working on it. Okay, so VFrog. So all the experiments which are there possible on the frog can be done with this VFrog, which is freely available on softwares in the computer internet. Okay, net पे आप देख लेंगे तो then dry lab plus fetal peak CD-ROM, then cocoon rat. Okay, like that we are handling a rat itself. That type of cocoon rats is also available. Even that is a dummy rat. Cocoon rat is a dummy rat. You can hold it properly. How to inject the drug into the tail vein. Tail vein में कैसे आप करेंगे? Rat के orbital से अगर आपको blood लेना है, which is very difficult and not permissible nowadays with the CPCA hard guidelines or rules regulations. Okay, so because of that, this cocaine rat can be utilized here. Okay, so if you see, there are from the old period, that you can say the 30 years ago or something like that, okay, or 50 years ago, so we used to have such type of laboratories which we have seen in the books and movies. Okay, and they used to go for this in vitro test on the animals. Okay, but and that used to take a huge time of 10 to 20, 30 years. But nowadays, what we are doing, we are having a targeted drug discovery system. Target identification is there. Genomics, proteomics, functional genomics. Then if we go for the target, specific target validation, protein expression assay development, then we go for the lead identification, lead optimization, and then directly go for the biological activity. And this can be enhanced. The number of years can be reduced with the modern technology. And here comes again, in this we are getting eradicating, you can see here, the in the animal testing with artificial intelligence. This was one article in 2018. The source is already given, that yes, we can minimize or we can remove the animals from the experimentation part to more extent. It cannot be completely removed by alternative method also, neither it cannot be completely removed by the artificial intelligence. Okay, and here read across structure activity relationship uses this binary fingerprints and Jaccard distance to define the chemical similarities, like what we have seen in Schrodinger जो है उसमें they have studied so many huge data which was there from the different publications, renowned publications, जो बहुत ही अच्छे अमेरिकन केमिकल सोसाइटी से जो renowned publications है, उनके सबका data, they have collected all the huge data, and because of that they have taken the help of the computational scientists, and with that they have developed the software. And now we can see that yes, for developing 5 to 10 good actual lead compounds, we can do it on computers within a 3 month to 4 month maximum period. But getting that 3 to 4 very good lead products in the earlier days, they used to take minimum 5 to 6 years. Okay, total PhD was used to complete, and then we will get yes, 1 or 2 compounds were there. But at least here, to maximum extent, we can shortlist the candidates which we can synthesize. Okay, which will have similar activities, which will have mimicking activities as that of the. So these softwares are very, very useful. Okay, they are constructed with the help of large chemical similarity adjacent matrix from the similarity matrix, and it is used to derive the feature vectors for supervised learning. Okay, with a given list of chemicals, it predicts whether a chemical is toxic or not. Okay, it can currently predict this for the tens of thousands of chemicals. And according to some research papers, These tests are consumed 57% of animals for the toxicological studies in Europe. Okay, so earlier there used to be huge quantity of animals which was used for this animal activity, but now it has been reduced to great extent. At the last phase only we can go for the animal activity. अगर आपको कंप्यूटर पे ही आपके सिमुलेटर मॉडल्स पे ही पता चल रहा है, okay, why need to go for animal activity in the starting itself? Okay, and there are promising results with this um, new concepts. So there is another article here. Artificial intelligence beats animal testing at finding toxic chemicals. So how machine learning could be the key to reduce the use of animals in experiments. And this study was carried out in the Hopkins University. Okay, and in 2016 they have compiled the data of about so much 8 lakh toxicological studies they have studied, and more than 10,000 chemicals are registered in the European REACH legislation. Okay, so in for the specific industrial chemicals which are used, okay, without setting foot in the animal lab. किसी भी animal lab में या pharmacological activity के लिए किसी lab में जाने से पहले ही इतने सारे chemicals, so much huge quantity of chemicals they have derived, and this is the science that yes, these chemicals can be toxic to the animals. Okay, so machine learning tools are very widely used in such type of things, and artificial intelligence based on big data set of toxicants is actually better than the animals uh, in the identifying the toxic substances. Okay, so John Hopkins University has published this report. And AI news, artificial intelligence, we are always saying that yes, it is developing. So in your earlier lectures itself, I hope much more detail you must have studied. I'm not going into the detail क्योंकि आपको फिर से उतने डेप्थ में ले जाके मैं नहीं करना चाहता हूँ. Even I'm not that much expert, we are also learning the things because that is need of an hour. So AI assesses breast risk cancer 30 times faster. जो एनिमल एक्टिविटी है या बायोलॉजिकल एक्टिविटी या दूसरी और जो अल्टरनेटिव्स मेथड्स है या टेस्टिंग किट्स है, और in spite of that also, this is because of the use of artificial intelligence. 30 times faster detection of the breast cancer is possible. Okay, GE reborn as a software startup using artificial intelligence. GE is the world renowned company. Okay, and it was about to finish a few years before, but they have changed their version, they have changed their structure and gone completely on software based type of thing, and especially using this artificial intelligence, and they are going like anything. Okay, otherwise if we have seen that there are companies which do not change themselves, okay, are going to abolish. The people, scientists or educators who are not going to change will remain stagnant at certain point. So to avoid this stagnancy, I request you, all of you, to get yourself updated, because we have seen the classic example of Nokia. Nokia एक जमाने में एक शहंशाह माना जाता था मोबाइल फोन के लिए. So in India especially, when phones started, mobile phones or handsets started, Nokia was the key. If we consider the example of photographic films, Kodak was the king throughout the world. Fuji film was the another world leader in the film. But all of a sudden digitalization came. Okay, कोई भी normal camera with film nobody is using now. Everybody is having digital cameras or mobile phones for digital picturing with very good quality. So Nokia has gone, Kodak has gone, film has gone. They have not updated themselves. Those industries who have updated themselves, who have utilized the latest technology are surviving and growing faster. Okay, and that is how the world is leading 2025, especially China in AI industry. Okay, so China is leading thing in the AI industry, and now here again picture comes is that India always wish to have a somewhat similar treatment with that of the China, especially after this pandemic. World has also seen that how they are doing their businesses and other things. So everybody is looking after India, especially pharma market, pharma industry. Okay, and now we have shown that within a few months itself, okay, we can become a good exporter, manufacturer as well as exporter for the PPE kits, masks. We were using that N95 mask in the biological rooms only, जहाँ microbiology के लिए वहाँ use होते थे. Doctors used to have only surgical mask. N95 was nowhere, but now we are producing so much number of N95s that we can export it. PPE kits we are not having it. We used to import it. Now we are producing it. So if we are updating ourselves, okay, we are going to survive as well as growth also is possible. So there are billion industries there for this, especially artificial intelligence. Okay, so in NASA they are also using voice recognition 3 times faster, self-driving taxis. Okay, in Singapore it is available also. So especially Deep Knowledge Ventures appoints AI artificial intelligence like a board member to make investing decisions. Okay, so आप लोग काफी बीच में अभी देख रहे होंगे, रिस्क है तो इश्क है, जो हर्षद मेहता की काफी सारी चीजें आप देखते होंगे कि that lot many scams was there in the stock market and other things, and because they used to play like a game with the stock market, but वहाँ अभी यहाँ इवन ह्यूमन ब्रेन की भी बहुत ज्यादा जरूरत नहीं रही है. Okay, they can, these softwares are with the help of artificial intelligence, they can see what the mood of the people who are working on the computers, उस पे क्या क्या mood है, accordingly they will adjust their decisions, and like crores and crore rupees can be earned with the help of artificial intelligence in the stock market also. Okay, उसमें काफी so many names are there. Okay, so robots, use of robots in medicinal as well as pharma industry is very important thing. So Baidu Ask Doctor has changed the way. Baidu Ask Stock Master. Okay, and the robotic experiments, self-awareness things, especially in the healthcare sectors, in neuroscience. Okay, this robotics as well as artificial intelligence is playing a tremendous role, and all of you, all of us, not only you, all of us uh, is needed that we should be updated. So in conclusion, I think, well, we must be careful in during conducting this animal experimentation, so that there should be minimum pain to the animal whenever needed. Okay, animals should not be used at all if any equally suitable model systems is available for the same experimentation, and every possible steps must be taken to reduce the or to prevent the pain and suffering to the animals. By evaluating the each research, teaching or testing protocols, adherence, the principles of 3 R's, okay, should be followed properly. Okay, and use of animals in the research or teaching, testing, you cannot see as our right, but it is a privilege. It is a privilege given to scientists or teachers like us, that yes, we can use these animals properly for the uh, training and research purpose in the pharmaceutical or medical institutes. Okay, so although there are efforts in the researching as well as alternatives to the animal testing methods throughout the years, okay, and successful results are also there. So many good softwares are available, and there is lot of scope is available, especially in the field of artificial intelligence, and that will be the key for further development. जहां आप देख रहे हैं कि फार्माको विजिलेंस जो फील्ड आया है, okay, so that field we are utilizing it properly. All the data, what are things are there, and depending on that data itself, okay, they can see that yes, what type of teratogenicity will be there, और क्या क्या चीज हो सकती है, conclusions क्या निकलते हैं, okay, they can do it on their own. Decisions can be taken by the systems with the help of artificial intelligence, especially. Okay, I hope uh, I'm able to justify the topic to some extent, because this field is very vast, like an ocean, and I have only added a drop in that ocean. Okay, I have taken a drop out of it, and if you do that small efforts at your level, okay, we can do a wonders. What we are seeing, like what mm, I can tell you with my experience itself. SGSITS Indore was a very small entity when I joined there as a PhD candidate in 1997, 99. Okay, that period, very small department, but the starting phase was there. Spark was given by Professor Chaturvedi sir that yes, we are going to start the drug design lab in the institute. Okay, and started, and there are so many good scientists, Suman, Subhash, Dr. Arun. They have started working properly, very nicely. Okay, and teachers like Meena madam, Neha madam, and Chaturvedi sir, Trivedi sir, so many good teachers was there, and they have developed. Students has taken advantage of the CDRI Lucknow and developed their labs to such extent that there is a great name for that particular department. Yes, they are good. They are masters in the drug design. Okay. In the same way, I suggest all of you to take some initiative at your institution level and start working on it slightly. Also, if you do the things, we can do wonders, especially in present scenario that is needed. Because if we go hand in hand with the IT persons, engineers, doctors, okay, in development of such softwares, use of this uh, artificial intelligence, we can develop very good number of softwares, especially because we are seeing that in most of the medical colleges, most of the pharmacy institutes, this even TCS are recruiting the people from the pharma background. SCL is recruiting. Okay, Cognizance is recruiting. So there are so many IT industries are also recruiting because they need in the engineers, but for studying these biological things, they need the good quality pharmacist also. And as we are from the pharma fraternity, it is our duty to train our students also, to train ourselves also, and the training or adaptability is much more in the students. So what I'm presenting here is a small token, but my students are much more faster than me. I am proud of that. So I request you, all of you, to get yourself updated. And I thanks again Dr. Arun Gupta and his entire team that they have selected such wonderful topics of artificial intelligence. Okay, I wish all of you a very uh, good futures in the field of pharmacy as well as artificial intelligence. Okay, and wish that in future we can meet. This is again a good thing that through online mode, so many I'm seeing the delegates from various organizations, very senior faculty members, and by sitting here in Delhi, I'm able to connect with you people. This is also technology, but in future, whenever possible, that physical bonding is also required. So you are most welcome to our university, Dipsaru, which is the first pharmaceutical university of India. And Dipsar is a very old, renowned institutions, about 65-year old. Earlier we were with the Delhi University, and now we are with the Dipsuru University, and Professor R.K. Goel is our Vice Chancellor, who was the Vice Chancellor earlier with the MS University, Baroda, and before that he was the associated with the LM College of Pharmacy. He's a great scientist, pharmacologist of India. Okay, and we are honored to have him with us as a Vice Chancellor. And we are having D.Pharm, B.Pharm, M.Pharm, PhD, as well as now we are having Bachelor of Physiotherapy, Masters in MBA in Pharmaceutical Management, Masters in Public Health, Masters in Hospital Management. Okay, and planning to start B.Pharm in Ayurveda type of things also. So we have to upgrade ourselves, keep ourselves updated. So BS in sports pharmacy also they have started, and so much good. Earlier we were hesitant that how the sports pharmacy can be correlated with the pharmacy, but they have got such a huge infrastructure, machineries. Okay, कि जो फिटनेस इक्विपमेंट्स इंडियन क्रिकेट टीम के पास होंगे या इंटरनेशनल लेवल के ओलंपिक पीपल के पास में होंगे, सेम टाइप ऑफ इंफ्रास्ट्रक्चर दे हैव डेवलप्ड इन आवर यूनिवर्सिटी आल्सो. So you are most welcome to our university whenever we are also conducting this QIP program, and presently Dipsar is the chief coordinating cell for the AICTE QIP. Ask your students for to join us as a NDF fellow, National Doctor Fellowships, and AICTE scholarships are also available with us for PhD purpose. Specially send the good students, meritorious students. DST Inspire Fellows are also most welcome. University toppers can easily or directly join with us as a PhD candidate. Those who are having with MPharm background, teacher faculty members can join us as a PhD candidate QIP also. So I think I'm exceeding my time, and my friend Professor Bari is also waiting. Welcome, Bari sir, and thank you, organizing committee, especially Dr. Gupta, for giving me an opportunity to interact with all the delegates. Thank you very much.